One, you have a syllabus. I printed it for you. You can bring it back signed by your parents. I'll scan it and give it back to you as evidence that they've seen it, and I'll give you extra credit for it. So that's it for the syllabus. That's the second semester. Please take a look at it when you have during advisory, etc. Look how much we still have to cover. Okay? And think about that the next time you're like, oh, we have a snow day, or yay, we get to relax, and why are you giving me more work? Why are you giving me work? Why? Look at that list of things that are going to be on your end of course exam. And understand that we have to cover it all. All right. Now, you have we're going to go over this practice exam, and we're going to go over it by going over the content. And hopefully you've read chapter 10. I've given you a second, a third, or fourth chance now, right? It's what, the third week of January is, or second week? I can't remember. It's the third week of January, and I gave you these assignments the chapter 10 reading, probably the second week. So you've had 2 weeks. You've had 2 weeks with these assignments, and they're not that hard. So read a chapter. Take some notes, copy a chart, and analyze a graph. Those are the 3 assignments that people have not turned in. And so I hope you've turned them in by now. I'm not going to give you much more time. Are we clear on this? So get it done. In this retake stuff, if you read the syllabus at the very top, there will be one change in that there will be no late submissions, resubmissions, or test retakes. The logistics of the first semester and the result of those policies in the first semester have demonstrated a change in strategy, have demanded a change in strategy. So it will not occur anymore. So make sure you really have to start focusing here, people. Or we're all gonna get, mainly you, are gonna have some serious issues. All right. So there's 2 kinds of cells that we've talked about. And there's more than 2 kinds of cells, but there's a eukaryote, is that right? And eukaryotes have nuclei, they have Golgi apparatus, they have internal membranes. Oh, I'm sorry. Let me switch over to the. It's okay. Nobody cares. Sorry, I had to say it one more time. Oh man, why is this? So when we're dealing with these cells, they have internal membranes. They have, and one of the things that we talked about, I don't know, forever, and still some people make mistakes, is this idea of a nucleus. I don't know how you can still make a mistake after this. And the nucleus has all the DNA in it, and the DNA is unwound in a form called chromatin. And these are eukaryotes, right? We've talked about those guys. And we've also talked about these things called prokaryotes. They don't have any internal membranes. Months ago, I think it was, I described the prokaryotes as shacks and the eukaryotes as houses. And I said the eukaryotes have rooms, many rooms, compartments, right? And that's why it's a fancy mansion. And a prokaryote is this single-celled organism without any internal rooms. Is that correct? Yes? Yes. All right, so that's a prokaryote. And to be clear, both of these have DNA. Both of these cells have DNA in the nucleus. It's just that the prokaryotes don't have any nucleus. They just have their DNA's in the cytoplasm. There's no membranes in the cell at all. There's just a plasma membrane, the outside cell. And of course there's a cell wall, right? Yes? I'd like to get some feedback from you here. Yes? Okay, very good. So there's some cell wall. It's on the outside of the cell membrane. Cell wall helps it protect against lysing because of osmotic changes that we discussed. Right? Yes. And it doesn't have a central vacuole because it doesn't have any internal membranes. Yes. Pay attention, sit down, pay attention, take notes. So I mean, I don't know how much more I could say, how many more times I can repeat myself to say, again, you need to know that. Is that clear? All that stuff I just said, you need to know it. And people were like, how come I got this question wrong on the test? It's because you didn't know that. You got 2 questions wrong on the practice test because you didn't know that. Now, of the eukaryotes, these guys here, there's several kinds. Now, of the prokaryotes, we've discussed already that there's 2 kinds. What are the 2 kinds of prokaryotes? I heard that, yeah. The prokaryotes, remember, it can be spelled with a K or C, depends on what they do. It's bacteria. That's right. I heard that. And Archaea Bacteria. Archaea Bacteria are extremophiles. They're pretty much single-celled organisms without a nucleus, so that's why they're prokaryotes, and that's pretty much it. They do different chemistry. There's some internal differences as far as how they do what they do, but they're still single-celled, no membranes inside their cell membrane, prokaryotes. All right? So, but for all intents and purposes, bacteria, Archaea Bacteria, you wouldn't know the difference if you saw them underneath a microscope. All right. Eukaryotes we've broken up into two types. We got plants and animals. Those are the big types, but there's more, aren't there? What are some of the other types of eukaryotes? 2s. Fungi, that's right. Which yeast are a fungus. Yeast are a fungus. So it's good that you remember yeast, but it's a type of fungus. And so what else? Animals, fungi, plants, protists. All right. Now these 3 here, fungi can be single-celled. They have spores. They have, let me think, are there single-celled fungi? Mildew maybe. There's different forms. Fungi are complicated. But we call these multicellular. What does that mean, multicellular? Yeah?
It means the organism is made up of more than one cell.
That means they're made up of more than one cell. So that would be, a human, right? Right? A meerkat, a mushroom. They're made of more than one cell, right? Think Luigi and the mushrooms and the stomping on them and all that, right? So those are multicellular organisms. That means there's more than one cell. How do they grow? Yeah.
Mitosis.
Well, what are these? These are all eukaryotes. So they grow internally. The cell gets a little bigger, but then what happens to the cell? They split, they replicate. They do the cell cycle. And what's necessary is mitosis. So mitosis is the process upon which they separate their nuclei because they have nuclei. Is that clear?
Yes.
But what about the protist? It has a nucleus. The protist has a nucleus. We're talking here, what are protists? Amoebas? Right? Those are those weird looking things, Osmosis Jones kind of looking things. They just move by changing their endoskeletons.
So amoebas are single cell?
Single cell, yeah. Amoebas, ciliates. Those are cells with cilia.
That means they can't perform mitosis?
No, they have to do mitosis, don't they? Because they have a nucleus. They have to go through some form of cell nuclear replication and splitting, right? Mm-hmm. But do they use that for growth?
No.
Why not?
Because they can't get bigger if they're single cell.
Yeah, they're only one cell.
Yes.
So they're not using mitosis. You went from one cell to a trillion cells because you're multicellular.
Yes.
Right? A single cell splits, if a protein splits, you had one, what do you get now? 2. That's how they replicate. That's how they make more of themselves. We, those of us that are multicellular, do something of, I mean, we might do asexual reproduction, but in general, it's sexual reproduction, right? We replicate by taking a single cell and multiplying it and growing, but to do replication, we have to create sex cells. Protists don't do that. They just split. They do mitosis and split over and over again, and protists will increase their numbers, right, their population by mitosis, but they don't repair tissues, right? Why don't they repair tissues? Using mitosis. We repair. If I cut you, how do you correct it? How do you heal? Mitosis. The cells start doing mitosis and cell cycle and replication, and they make more cells, and they replace the cells that died. Why are protists not doing that? Because they're single celled. They don't have tissues. That's it. That's all there is. It's one cilia, one amoeba. It splits into 2 amoebas. It's split into 4 amoebas, etc. Is that clear? That's a question a lot of people got wrong on that practice test, and there's absolutely no reason for it, is there?
No.
Right? If you think about it, just think. And that's the hard part during a test, especially when you're under a lot of pressure, isn't it? Right? It's the thinking that becomes difficult. It's not that you're stupid. It's not. It's not that you're freezing or you have some kind of anxiety problem. Look, everybody, it happens to everybody. When you go in the armed services, and you go to the Army, Navy, and Marines, they put you through something called boot camp and AIT. Why do they do that? Why do doctors have to spend, at least in the old way of preparing doctors, why do they put doctors through sleep deprivation and stress and yell at them and put them under a lot of pressure during medical school and training? Why do they do that? Yeah.
It's kind of like conditioning for sports.
It's very much like conditioning for sports. They're trying to prepare you for what happens. Why do I stress you out on practice exams?
For the big test.
So that you can get ready mentally for how are you going to deal with that stress when you're sitting there having a question like this one and you can't figure it out and you panic and you choose the wrong answer. Stress is necessary for conditioning. Now, where are we? Everybody gets this? Yes? Questions? I thought I saw some hands up. No? All right, someone close the door. Now, let's go a little bit more, let's talk a little bit about meiosis that you should have already read about. You've done a flip book on, and some of you have done amazing little flip books. All right. So, let's just look at the nucleus. So this is cell, we're going to draw a cell here. There's a nucleus, nucleolus, there's an endoplasmic reticulum, Golgi body, vesicles, and mitochondria, and whatever, right? All kinds of organelles, the RNAs in the nucleolus, and they got the DNA, chromatin, etc. We know the cell cycle. We got G1, there's G0, right? And G1, and then there's synthesis, and then there's G2, and then there's the M phase, is mitosis or meiosis, and then back to G1, cell cycle, doing cellular replication. All right, everybody should know that. That's fine. Let's keep that in mind as we go on. Yes? Now let's do meiosis. Mitosis, as we discussed, if we have mitosis, we have a single, and everybody here, I think, understands the basic concept. Once you've made a copy, you have this chromosome, and it looks something like this usually. 2 strands of DNA, copies of one another, yes? Where did you copy this? In the nucleus, in what phase? Interphase is correct. What part of interphase? S, synthesis. You copied it up here. That's what S stands for, synthesis. I would expect that people are writing it down, right? Because I didn't hear any right answers. I heard 0 right answers when I asked when is DNA copied. This is important that you know this so that you understand why meiosis is So special. Synthesis. That's where DNA is copied. So, as this DNA was copied here in synthesis, but it didn't condense. You couldn't see the chromosome until prophase. That's when you started to see it. Why would you? Because it's the end phase when you're ready to split the nucleus. That's when you have to pack everything up. All right, let's say you're going to move in 5 years. Are you going to pack your stuff up now? No. You need to have your socks, your underwear, your shirts, your books, your papers, your shoes. You can't put all that in a box, and then what are you going to do when you need it day to day for the next 5 years? It doesn't work. So when we wrap this up, when our cells wrap up the DNA and after it's been copied and connect them together, we're getting ready to move them. We're packaging, we're putting them in boxes. Are we clear on that?
Yes.
Just like when you're moving from house to house, when you're separating this nucleus, we want to make sure everything goes where it's supposed to go. These two things are called what? This one and this is both, let's call them both chromosome number 1. They're both they're copies of one another, so they're copies of chromosome number 1. What is each individual called? Yeah.
Sister chromatid.
Sister chromatid. Everybody should know that. If you I don't know why you would not know that. I really am confused if you're sitting there going, I didn't know that. Why? We've covered it in class, you've read about it, or you should have. Sister chromatids. This is again one that surprised me that people got wrong on the practice test. Unfortunately, so these are copies, yes?
Yes.
Exactly the same DNA, except for maybe some errors. There might be a mutation in there, but we're not going to discuss that right now. The bottom line is these two are ideally, sister chromatids are exact copies. But there's 2 chromosomes in there. 2 chromosome number 1s. One came from mom, that's this one here, that was copied, so both of these are exactly the same genetic code. And where'd the other one come from?
Dad.
For the other one came from dad. And that again is chromosome number 1. Now these are both chromosome number 1, and of course this one's identical to this one, right? Are we good with that?
Yes.
So these 2, this one came from dad. What do we call these 2? They're both chromosome number 1s. What do we call them? Not yet. We're not at the sex cell state. We're not there at that discussion yet. Yeah.
Is it a homo?
They're very good. Excellent. Excellent. God. Oh, some kids, they just saved my sanity. Yes, it's homo. Remember, homo means what? Same. So this is homolog. All right, so homologs. Homologs are the same chromosome. Are they identical? No, they're not identical. Your dad's genes obviously are slightly different from your mom's genes. Otherwise, they'd be clones, right? And that's no good. They'd be twins. And if your mom and dad are twins, you got problems. I don't even know how that would work. But the bottom line is that, anyways, chromosome number 1, they have the same copies. We're going to say they're homologs, okay? That means that they're the same genes. If that genes for eye color is here, right? If the gene for eye color is here, then it's here, and it's also here, and it's also here, right? So there's 4 copies. Do you agree? At this stage, there's 4 copies of the gene for eye color. Now, what's the difference? Well, the difference is that if this gene for eye color is A, big A, then this one's probably big A too, isn't it? Because they're exact copies of one another, yes?
Yeah.
However, dad's doesn't have to be a big A. Dad's could be a what? A little a. That's the difference between homologs and sister chromatids. Sister chromatids have exactly the same DNA, but homologs have the same genes, but might have different sequences. This was, again, some questions on that practice test you guys did not do well on, in general. I mean, 70 is okay, but I was expecting 100%, to be honest. All right, are we good with this? All right. So that's the basic terminology we've already covered. It was in the chapter, but some people messed up and for whatever reason didn't get it straight. So now let's go on to the next thing, mitosis. All right. Mitosis, you're taking one cell, and what are you doing with that one cell? You're replicating. You're making two. But specifically, mitosis is not the cell replication, right? Mitosis is what? The nucleus, right? The nuclear replication, the splitting of the nucleus. The cell replication is at cytokinesis, which is a different step in the cell cycle. The cell cycle is the life cycle of the cell. So a lot of times you'll hear people talking, even people like me that are supposedly teaching the course, will make the mistake of saying mitosis when we really mean cell cycle or cell replication. Is that clear? So you got to keep that mind, that you straighten your mind. You'll hear professors say it, you'll read it in books that way. It'll seem like mitosis is being used as a synonym for cell replication. But mitosis officially is the splitting of the nucleus. All right. So in mitosis, we have to make exact copies. So if we have, what do we have in a regular cell? Let's take a look at what we have in a regular nucleus. So if we look in a nucleus, we have one chromosome from mom, one chromosome number 1 from mom, right? And we have one chromosome number 1 from dad. I'm just going to use just this one chromosome to keep things simple, all right? So, and this one's from mom, and this one's from dad. Just that easy. All right, so this one's big A, this one's little a, this one's big B, this is little b. This is big C, and this is little c. All right, so dad has all recessive traits on that particular, for those 3 genes. Remember, there could be many, many genes in that chromosome, right? But for these 3 genes, dad is recessive for all 3 of them, and mom is dominant for all 3 of them. So you have these, what did we do during S phase then of the cell cycle? We copied the chromosome. So we have this chromosome was copied. We connected it with the kinetic core, right? Right? Yes. Centrosome, right? And it's an exact copy. So there's A, there's B, there's big C, there's little a, little b, little c. Again, this time, of course, separate chromosome. Are we good with that? All right, so they're copied, you have them, now you got to split them. So now at prophase, that's what they look like, but what are you going to have to do if you're going to split them? I mean, if you think about it, everything in mitosis makes absolute perfect sense. If you think about what cells have to do, they got to move them. How are they going to move these to the cell, to the molecules that make up the cell? These chromosomes are humongous, right? They're 3 meters long. They're wrapped up around proteins called histones. They have these big shapes. They're unwieldy. They're difficult. Imagine the pyramids, building the pyramids. So moving these chromosomes is like humans building a pyramid. What did they need to build the pyramids? Lots of people, correct. Yeah. Well, what did they do? How do you move a giant stone the size of a house?
Sleds. They use large sleds.
Sleds, and what else?
Pulley.
Pulleys. You had to use ropes and pulleys. You're not going to have how many hands can you get around a stone like that? A bunch, but is that enough hands to move a stone like that? No. You need ropes and pulleys and all kinds of levers and mechanisms. So if you're gonna move these giant unwieldy chromosomes, again, giant as far as the cell molecules are concerned, then you need those ropes, and we call those ropes spindle fibers. And those spindle fibers, what they do is they kind of form this kind of thing from one end. Now, it makes sense that they're from one end to the other, doesn't it?
Yes.
Does it? It makes sense because you're splitting the cell. So you got to pull the cell, the chromosomes either to one side or to the other side. right? So it makes sense that they're laying out, they're basically laying out the side to one side and then the other, right? Right and left. I mean, that makes sense, up and down, whatever, but one side and then the other, two sides, yes?
Then, if we're getting the same chromosomes from our mom and our dad, if I'm understanding this correctly, then is it the mutations are the reasons why, like, my siblings and I, even though we all have the same mom and dad, that we all look big?
Let's hold on that till we get to meiosis, all right? Again, I'm going to say something, and I don't mean it as kind of, I don't mean this to be snotty, all right? But I'm just going to say, read the chapter, all right? It's poignant what's written there in both copies of chapter 10. Remember, chapter 10 is mitosis and meiosis for both chapters. All right, so one side or the other. Right, so it makes sense that now you got to have a way to connect the chromosome to the spindle fibers, and that's these kinetic cores, these centrosomes, right? These centrosomes are the way you connect how you move it, right? But wait a minute, a bunch of ropes on the ground aren't going to help you move these things. How are you going to make sure you get them? Yeah.
Oh wait, I think I was.
Go ahead, say whatever you're going to say.
Oh, I was going to say it was attached to the central.
That's right. You need to have them attached to something, right? An anchor point. And in animal cells, you need these things called centrioles. You don't need these things in plant cells because what do you have? Cell walls, right? And I'm just kind of sketching them out. So all these spindle fibers that attach, now they're all secure, and you got them attached to your chromosome, yes? So now what? If you're going to split them, do you care that you get a mom's copy on this side, and right? Because if you want exact copies, if you got one from mom, right? Let's just make mom's. You know what? Let me make mom's red, and one from dad. Let's blue. Let's go ahead and be gender specific, right? And if you got one red from mom and one blue from dad, right? They're both the same chromosomes, just different alleles. So, is it okay that I do this then? No, it's not okay, because are they identical? No, because this cell here would be big A, big B, big C, and this cell here would be 2 little a, little b, little c's. This might be diseased, or this might be, who knows? This is not okay. If this is the cell that you have and you want to copy it, then you have to have both mom and dad's in both cells. And that's where it becomes important that why mitosis happens the way it does, which is that the chromosomes line up in the center during metaphase, right? And I'll go ahead and draw one chromosome red and one blue chromosome, just so that we can look at it here. That's why it's important that you do this, that you line them up this way. Why is it important you line them up this way? Because what can happen if you do it this way? Think about it. I'll wait for an answer.
Oh, the sister chromosome.
You can split the sister chromatids. That's right. Because by lining them up this way, what are you going to get when you split them?
Like half going to one side, half going to
Right, exactly. This one goes that way, this one goes this way, this one goes that way, this one goes this way. When you're done then, and at the end of mitosis, you're going to end up with one blue chromosome, right, and one red one in each cell, in each nucleus. And in both of them, you're going to have one red one and blue one. In other words, you have an identical set. But that's because, why is that? Because you line them up this way. And what did you split? When you did anaphase, what did you split? Remember, anaphase is a splitting of the sister chromatids. What did you split? This at the centrioles, using the kinetic cores that are in the centrioles. What did you split?
The chromosomes.
The chromosomes, what do we call them when they're together? The copies are together. You split the sister chromatids, right? When you're talking about this, what are you splitting? When this was going that way, this was going this way. The two sister chromatids are splitting. And you're right, they're chromosomes. The sister chromatids are chromosomes, they're copies of each other. We just call them sister chromatids. Is that clear to everybody? All right. So we split them, and we get identical copies. We get A, big A, little a, big B, big C, little c in both cells. Because we split these two, and we split these two, and one set in each cell. That's mitosis. That's really the big idea there. The whole point of mitosis is making exact copies, two cells with exact copies. Now let's go on to meiosis. Now meiosis is a little different. Yeah.
Doesn't cytokinesis come after?
Yeah, cytokinesis comes after anaphase, after they split, you get the telophase. What are you doing? You're reforming the nucleus. Don't forget, during prophase, you got rid of the nuclear envelope, because you need to be able to split them on two sides of a cell. You can't have the nucleus getting in the way, so they get rid of the nuclear envelope, right? Now, in telophase, the chromosomes are on opposite sides of the cell. The spindle fibers start to break up. You don't need them anymore, and you start to reform the nuclei. That's why during telophase, you're going to see how many nuclei usually? 2, right? And then in cytokinesis, you start to see the pinching off. The pinching off of the cell in an animal cell. You'll see the pinching off of the cell because the cell has to pinch off to become 2 cells. In a plant cell, you'll start to see a cell wall form. We're going to do a lab this week. We're actually going to look at these things under a microscope. Is that clear? So you're going to have to count how many cells do you see in anaphase and metaphase. Do I really care that you know the steps of metaphase and anaphase? Not really. What I care is that you understand the process so that you get the idea that in mitosis we have 2 exact copies, yes? And how and why is it important that they line up in that metaphase plate? And that the two set, and that's going to become, I think, clearly important here in meiosis. And one of the big differences between meiosis and meiosis, you're going to see in a second, is going to, I hope, accentuate the point that splitting the sister chromatids keeps the cells the same, and not splitting them makes them different. And that goes to your question you asked earlier, which is what is it that makes me differ from my sister? All right. Yeah.
So we can never feel this happening.
Feel it happening? Huh. No. This is all in the cell. Remember, you feel because of nerves. So your nerves have little inputs, tiny little inputs that add together, that signals that add together, and your brain interprets them as pain, pleasure, light, and sound, and smells. All right? So the tiny little molecules are floating in the air. Enough of them hit your nose. They send tiny little signals of electrochemical changes throughout an axon and dendrites into your brain. And your brain says, if it's enough, your brain's like, oh, that's an interesting smell. You smell millions of things all day long. You're just like, ignore it. It's not enough. It's not important. You cannot process everything, yes? You can't process everything that's going on in the world. It's too much. That's what happens to some kids that have autism, right? There's too much information. They can't, their brains can't separate it, and all they see is white noise. It freaks them out. I don't blame them. My nephew has autism, and you can't touch them. It's too much information. Their brains, it's an overload. Okay? So to answer your question, no, you can't sense that because this is at the cellular level. So it's a different level, right? It's deeper. This is all molecules moving around. You have to be able to have a nerve ending to sense it. You can't sense it because it's inside the nerve. It's below the level of nerve, right? Smaller than the level of nerve. Not to say less important, just to say it's smaller in size. Okay. So meiosis again, What's the point of meiosis? Why do we do meiosis versus mitosis?
Sex cells.
To make sex cells. And so we need sex cells. There are two types in humans. In plants we call them the sex cells are things like pollen, right? And in chickens they're chicken eggs. In female humans we call them eggs. In male humans we call these sperm, right? They're not round, but they're kind of more like they're more oval. And the female has an egg, but what's so special about these two cells? What makes sex cells special? Yeah.
The sperm cell is a diploid cell and the egg is a haploid.
Nope. Yeah. Genetic variation is definitely one. So these two cells are different. Let's just put the word different. Different how? Genetically. Right? Genetically different. We all agree? So they're different genetically. All right? So that's important. But there is something, as someone mentioned, haploid diploid. They said it incorrectly, though. They said that the sperm cell is diploid and the egg cell is haploid. That would be no good. That would lead to real big problems.
It's the opposite, that's why.
No, it's not the opposite either. It's neither one of those. Yeah, they're both haploid. So they're both half the DNA. They both have half the DNA that a body cell would have. Half the DNA. If you think about it, it makes absolute sense. You're trying to make one whole. How do you make one whole person? You bring two halves together. If you bring a whole and a half, that's not one, that's 1.5. Right? That's no good. A one and one makes 2. But half and half make 1. Come on, guys, you know this. It makes plain sense, yes? All right, so half and half is going to come together and make, but how do you get half? How do you get half, and how do you decide what half? And how do you make things genetically different? See, that's the details of meiosis. Which is why I had you make those flip books. And I saw you draw the chromosomes. Did you not know what you were drawing? Because your answers on your practice test did not match what you drew. Thought. Deep thought. Reflection. Summary. Staying awake in class. These are all important things. Yes? Right? Yes?
Yes.
Okay. All right, so let's go back. Give me a second. So I copied this because I want to say, look, this was mitosis, but we're not going to want that in mitosis because you see how they have 2 copies here, right? And we have 2 cells. We need to half it, so we're not going to do that. And let's see, we are going to copy it. We are going to do the S phase. That's correct. So we are going to copy it. We are going to do S phase, right? We're going to do meiosis, so this is part of the M phase. We have to grow the cell, so we have to do the G1 and the G2. So all that's still going to be the same. The first round of meiosis, meiosis 1. Mitosis is just meiosis meiosis is just mitosis twice with a little twist. That's all. So if you understand mitosis, meiosis is easy. So one of the problems that you see here is that this is not what mitosis does. So the meiosis rather. We don't do this in meiosis. So that's a little different. They don't line up that way. We still want to move them from one side to the other, right? But we're not going to line them up that way. They are copied. The homologs are copied, but they do something really interesting. They form something called a tetrad. Right? And if you read your chapter, you know what that is. But tetrad, what does tetra mean? Anybody know what tetra means? Yeah, like Tetris, yeah?
Build up?
Nope. Means four. Four. Tetrad. Why do we call this a tetrad?
Because that's four sister chromatids.
Four sister chromatids. It's not hard. Really isn't. So you have four sister chromatids, we call them tetrads. Let me make this line a little thicker.
I have a question.
Yeah.
So if it means four, then what? Like, does it have to do with the word Tetris, kind of?
I'm not sure. Ask the Latin or Greek teacher.
That's like so much work.
I know. If it's too much work for you to ask them, maybe it's not important enough to ask me here. What do you think?
You can ask them on the way to
I
school.
respect
You can
that.
pass them
Yeah,
on.
I'm just saying. It's like if it's not important for enough for you to look it up, then let's.
It doesn't. I'll take my.
All right. So
Thank you.
they line up this way. What is it? What's the difference? Yeah.
When like they move and Anaphase take the whole.
That's right. Excellent. Because somebody did their homework. They did the flip book. They know that when this one moves from one side to the other, they go together. So now you get both dads' blue copies here. Yeah? And you get mom's in the other one. Right? And remember, they're not going to form a sperm and an egg. So don't get that confused. They're either going to form a sperm or an egg. Is that right? Depending on if you're a male or female. Is that correct? Yes? I don't hear you. If I don't hear you, I'm not sure if you understand. I can't read your mind. So these have to form. But what is the big difference right now? Right away, after meiosis 1, we've separated the sister chromatids together. So we've kept moms and dads together. So now this cell has all of mom's DNA, and this one has all of dad's. Now, they don't move that way. You might get mom's chromosome number 1 here, but maybe you get dad's chromosome number 2, okay? And maybe you get mom's chromosome number 2 here, but then you get mom's chromosome number 3. You see what I'm saying? So they separate, each of the chromosomes separate, but they stay together, but they're randomly assorting, the homologs are randomly assorting. So that's where you get some mixing up of the DNA, right? Where you normally would have had, like, which chromosome number 1 did you get? Which one did your sister get, right? Who knows? Maybe you got chromosome number 1 from your dad. Maybe your sister got chromosome number 1 from your mom. Maybe you got chromosome number 1 from dad, and she got number 1 from dad, but chromosome number 2 is different for both of you. Who knows? We can't. It's all random. So that's number 1 source of genetic variation, is that these homologs separate randomly, but they separate together. So we've changed the DNA from the cells that your parents had to something different. All right. Part 2. During prophase 1 of meiosis, something weird happens. Anybody know? I know some people drew it in their little books. Yeah? Crossing over. And what crossing over means is something real simple. These are DNA, and they line up, and they're the same sequences. And the strange thing that happens is that these two can do what?
They swap like parts of
They swap pieces and parts of their DNA. They swap pieces and parts of their DNA. Very important. So now you got a little bit of chromosome number 1 from mom on dad's chromosome. And now you got a little bit of chromosome number 1 from mom on dad's chromosome on one of the sister chromatids. Notice the other one is what? It stayed the same. So now we got, where we used to have 2 sets of DNA, right? Big A, big B, big C, big A, little b, little c. Now we got how many different kinds of DNA here? Look at it and count. We used to have 2 reds and 2 blues. That's 2 kinds, right? 4 copies, but 2 kinds. And we have 4 different kinds now. We doubled the variation. We doubled the genetic variation, and it's random. So who knows what we made? I don't know. And how are they going to be separated? Which chromosome? This multiply this times 23, right? Because there's 23 pairs. So imagine doing this 23 times. Can you imagine the genetic variation? It's a lot. I could give you numbers, but let's just say it's a lot. It makes you different, more different than any other human being on the planet. There's no one exactly like you unless you have a twin, genetically at this level. Okay? So that's it. So it's called crossing over. It's genetic variation. It happens during prophase 1, during meiosis, and of course you have the independent assortment of the homologs also increases genetic variation. So let me erase this for a second, and let's update this drawing to include the results of crossing over, if you don't mind. That would mean I switch some of the DNA. Does everybody get that? We're switching some of the DNA, yes? Yes?
Yes.
All right. So because I don't hear you, as my drill sergeant would say. So we switched it there, yes? All right, so now what happens in meiosis 2? It's exactly what happens in meiosis 1, and mitosis. Meiosis 2 is there's no difference between meiosis and mitosis in the second phase of meiosis. The difference is that during the cell cycle, you go from M phase, you did cytokinesis, right? That's done. So you did M phase, you separated, but you didn't do mitosis, you did meiosis, so there's a little difference. This is the difference, right? You do have 2, this is meiosis 1. You go to G1, you start growing again, you gotta have the endoplasmic reticulum, all the other stuff that has to be replicated and all that in G1. Then what do? You go to S phase, but you're not gonna do S phase before you do meiosis 2. Do not do it the second time. You're not copying the DNA again. Is that clear? You're keeping the DNA, you don't copy. You don't do S phase the second time. You only did it the first time for meiosis. For meiosis, you only do S phase once. So what did that mean? That means when we split them again, when we do the same process all over again, you're going to split this in 2 more again, right? What does that mean? If we're going to do it again, we're going to make how many cells?
4.
Make 4 cells. 1, 2, 3, 4. They're going to line up along the metaphase plate, and this time you're not separating the homologs. What are you separating? The chromatids. Excellent. So you get one sister chromatid here. That's the blue one. One sister chromatid that has part blue, part red, part of mom, part of dad's. Another sister chromatid that has all moms. And another sister chromatid that has some moms and some dads, right? Yes? 4 different cells that have half the DNA. 4 different cells that have half the DNA. 4 eggs or 4 sperm. Depends on who you are. Now you put these together, and remember this is going to be what? Doubled, right? Because what's happening here is also happening in your spouses, right? In the person you marry, in their sperm and eggs, or whatever it is, depending on the combination. Their moms and their dads, their genetic combination. You can see how this leads to incredible genetic variation, right? Because they did it on their end as well. Now, which one of these sperm is going to find which one of those eggs? Who knows? And you've done this 10,000 times in the ovaries of a female human. And you do these you make 1 million copies, these 1 million genetic recombinations happening every time, all day, every day in a man's testicles, right? In a male human's testicles. So 1 million sperm are released during conception. And that's just one conception event, yes? So it's constantly being rearranged, the DNA is being rearranged. So that's the essence of your genetic variation. Include on top of that the mutations that happen. That's what gave you the alleles, the different alleles. then you can see how you can have such different genetic, so many people with so many different genes. So we'll go over this. I need my test back. We didn't get to the group base. You're going to be in the same groups tomorrow, and we're going to look at these questions on the practice test with this information in hand. All right, thank you.